Hi, everyone. For those of you who don't know me, I'm Steph. I'm a postdoc in Jenna Galloway's lab at Mass General Hospital, and I'm very excited to talk to you today about my work on tendon regeneration. So tendons are essential connective tissues. They connect and transmit forces from muscle to bone, ultimately enabling movement throughout the musculoskeletal system. They're extremely dense matrix tissues within ECM that is predominantly comprised of highly organized collagen 1 fibers that run in a highly organized manner from muscle to bone. And embedded within this dense collagen matrix are arrays of tendon cells, which we call tenocytes. Tenocytes are extremely elongated stellate cells with fibroblastic morphologies. And you can see what this looks like down here. So on the bottom here, I'm showing you, this is a longitudinal view through an adult zebrafish tendon. These cells are extremely elongated, and you can see that they're really just squeezed into the tiny spaces left available within this densely packed matrix as shown with SHG in gray. They also have stellate cell morphologies with extremely long processes that extend outwards and form gap junctions with neighboring cells, ultimately creating an intricate cellular network within this dense matrix. And both the cellular network as well as this highly organized collagen matrix are critical for force transmission and tendon function. So tendon injuries are extremely common and difficult to treat effectively. Some of you may have personal experience with a tendon injury, so you might know this firsthand. Here I'm just showing you some of the most commonly injured tendons across the body, including flexors and extensors in your hands and feet, your Achilles, as well as various rotator cuff tendons in your shoulder. The most common injury sites are typically in the middle, which we call the midbody, or at the tendon bone attachment site, which is called the enthesis. And I do want to highlight here that the enthesis is a very specialized structure at the interface of tendon and bone. It consists of a gradation of different cell types ranging from tenocytes to fibrocartilage and ultimately transitioning to subchondral bone. This is also paired with a very specialized ECM. And so this unique architecture at the nexus of tendon and bone is really critical for proper force transmission between both tissues without rupture. And unfortunately, after severe full tear injuries, for instance, in adult mammalian contexts, adult mammalian tendons can't regenerate. And instead, while they can heal, this is a slow process that results in fibrovascular scar formation as visualized here. So on the right, this is a mouse Achilles tendon 10 weeks after I performed a full tear injury through the enthesis, severing it off of the calcaneal bone in the ankle. You can see that the scar tissue is disorganized. It even has evidence of heterotopic ossification or ectopic bone now forming within tendon. And so this scar tissue is going to be impaired in function and more likely to re-rupture in the future. And current treatments are pretty limited for patients to non-operative options like physical therapy or surgical intervention, which is costly and still highly variable in efficacy depending on various factors. And so patients are oftentimes still left with impacted quality of life and a higher likelihood of potentially developing joint degenerative diseases down the line, especially with age. And better treatments are really needed. However, therapeutic progress in this area has lagged, especially relative to other musculoskeletal tissues, in large part due to the lack of an adult regenerative model that can be leveraged to understand how to mechanistically regenerate a tendon after injury. And so I'm going to summarize many years of work on one slide here, but long story short, our lab has really established the adult zebrafish tendon as a new regenerative model for the field. We've predominantly focused on a craniofacial tendon called the maxillary superficial tendon, or for short, the MST. It's located in this part of the zebrafish face, as you can see visualized on a Scleraxis M-cherry tendon reporter fish. Scleraxis is the tendon reporter gene that I just want to highlight here, and I want you to remember it for the rest of the talk because I'll refer to it many times. The MST connects the maxilla to one of the jaw adductor muscles. It's about 1 millimeter in length, and experimentally it's very advantageous because it's superficial, so it means it's easy to injure, it's easy to image, and it's also not required for feeding. And so if you injure this tendon, the fish is not going to starve to death. And so, like I mentioned, common injury sites include the midbody and the enthesis. And a prior postdoc in the lab, Marie, had actually shown if you fully sever the tendon off of the bone, that the enthesis can fully regenerate. And similarly, in published work, I've also shown if you sever the midbody, that the midbody can fully regenerate both the matrix as well as the cellular network. And so the adult zebrafish tendon is very good at regenerating different parts of the tendon. And so the next logical question is, well, what's the cell source of regeneration? regeneration. And we reasoned that potentially it was just the sclerosis positive cells themselves within the tendon. And so in order to address this question, I generated a sclerosis Cree-R lineage tracing line just to perform a simple lineage tracing experiment. This was performed in the zebra bow background, and so labeled cells will be CFP and or YFP positive. And so I pre-labeled the sclerosis positive cells prior to injury, injured, and then imaged to see whether they contributed to regeneration. And I hope you can appreciate in both contexts, you see a massive contribution of these cells, suggesting they are a major cell source of regeneration. So why do we care predominantly about this? Well, we care largely because we know that if you perform analogous lineage tracing experiments after full tear injuries at both of these sites in the adult mouse Achilles tendon, for instance, that these cells don't really contribute to scar formation. They kind of just remain dormant or quiescent. And so this really suggests altogether that divergent sclerosis positive cell responses after injury may be a driving factor for regenerative versus non-regenerative outcomes. And by studying and elucidating mechanisms required to activate tenocytes after injury, we might be able to inspire new therapeutic strategies to help patients within the clinic and improve healing outcomes. And just shifting a bit of the focus here, I do want to highlight that now we have identified a cell source that's capable of regenerating different parts of the tendon. And so this is a unique opportunity to actually identify whether there are injury site-specific regenerative programs that are required, and can we actually leverage this specific knowledge to tailor treatments within the clinic for patients for different types of injuries at different sites, for instance. And so we can also use that information to inform therapeutic strategies in the future. So that brings me to the major question that my talk is going to focus on today, which is a very basic question. How do tenocytes actually decide which part of the tendon to regenerate? And so in order to get at this question, I wanted to start with a pathway that I had already previously identified as being required for mid-body regeneration, and that was TGF-beta signaling. So in prior published work, I had shown if you inhibit TGF-beta signaling using a chemical inhibitor beginning immediately after injury or even delaying the onset to 1 day post-injury, you get a complete inhibition of regeneration. The severed ends fail to form a bridge that reattaches, and this is quantified on the right here. What was surprising was when we performed the same regimens in the enthesis regenerative context, the phenotype was less severe. So if I started the inhibition immediately after injury, most of them could still reattach to the bone in some way. And then if I delayed the treatment by 1 day post-injury, most of them reattached and actually looked quite normal at 7 days post-injury. And so this really suggested that TGF-beta signaling might be more selectively required for mid-body regeneration rather than enthesis regeneration, which begged the question of, well, what mechanisms are going to be driving enthesis regeneration then? And in order to sort of try to approach this question, we really needed to gain a better molecular understanding of cell populations both within the adult zebrafish tendon as well as surrounding the tendon, as well as a method in order to try to identify candidate pathways that might be driving differences at the different injury sites. And so I performed single cell RNA sequencing during homeostasis, as well as during various time points during regeneration of the enthesis and mid-body. We were able to recover many different cell types, including tendon and connective tissue clusters, which I'll focus on here today. I was really happy to see when I subclustered these cells, I saw a nice molecularly distinct population of enthesial cells separated from mid-body and myotendinous junctional cells. I also was able to recover 3 injury responsive cell states, which I'm not showing here, but actually are conserved between the 2 different regeneration types, which is interesting. And 3 tendon-associated fibroblast clusters, as well as other connective tissue subtypes as well, like osteoblasts and chondrocytes. And so using this data, I wanted to see whether these populations were evolutionarily conserved. So I performed cross-species mapping to mouse single cell data from the Achilles tendon, as well as human hamstring tendon single cell data, and was able to nicely see that these cell populations are actually conserved across zebrafish, mouse to human. And additionally, I wanted to further characterize the spatial location of these populations. And so I utilized this data to generate a 500 gene custom gene MERSCO panel and perform spatial transcriptomics. in which I was able to utilize the markers that we identified to identify the spatial positions of these populations along the tendon axis from the bone all the way down to the muscle. And I'm just showing you the core tendon populations here just to illustrate the point. And using this custom MERScope panel, I've also extended this analysis into a regenerative context. And I just want to, I don't have time to go into this in detail today, but please feel free to ask questions at the end. But I just want to illustrate here that using the markers that we've identified in this panel, we can actually identify the regenerating populations at very high spatial resolution. And so on the left here, I'm just showing you the enthesis regenerative context, and you can see the tendon reattaching to the bone here. And then in the mid-body as well, you can see the tendon ends reattaching, and that these are transcriptionally distinct populations at the regenerating area. But I just want to return back to the question at hand, which was, how do the tenocytes actually decide which part of the tendon to regenerate? And so leveraging all of this transcriptional data, I performed a differential cell chat analysis between both types of injuries, and identified several differentially active pathways, but one really stood out as being very highly differentially active, and that was canonical Wnt signaling. So here I'm just showing you two chord diagrams. On the left is the mid-body, on the right is the enthesis, and I hope you can just easily appreciate that canonical Wnt signaling cell-cell interactions are much higher on the enthesis injury context instead of rather than the mid-body injury context. And this really suggested that site-specific differential activation of Wnt might be critical for directing enthesis regeneration. So of course I wanted to go in and validate this experimentally. And so using a existing Wnt reporter fish, I was able to identify that indeed there is differential activation of Wnt with much more a higher level of Wnt responsive cells in the enthesis regenerative context at 3 days post-injury. Crossing this Wnt reporter into the scleraxis lineage tracing line, I did see that scleraxis lineage cells also become Wnt responsive post-enthesis injury. And I also wanted to probe the functional consequences of perturbing Wnt on enthesis regeneration. So I started with Wnt inhibition and performed global chemical and genetic Wnt inhibition experiments. For the genetic inhibition, I utilized a pre-existing line designed and generated in Gilbert Whitinger's lab, which allows DOX-inducible global activation of a Wnt inhibitor axon 1 upon DOX administration. And so Wnt inhibition globally from 1 to 5 days post-injury led to a significant increase in proliferation during regeneration. And this could also be phenocopied with a similar treatment regimen, just using the chemical inhibitor IWR1 as well. And so this really suggested that early Wnt inhibition might be beneficial and boost proliferation. So what happens when you hyperactivate Wnt? So interestingly, if performing chemical Wnt hyperactivation experiments using the canonical Wnt activator bio from 1 to 5 days post-injury led to the complete opposite effect. So it led to a significant decrease in proliferation. This also was coupled with a decreased recruitment of scleraxis lineage cells. And interestingly, if we actually hyperactivated Wnt even earlier, beginning immediately after injury, this was detrimental and actually led to more severe phenotypes, with the tendon not being able to fully reattach to the bone in many contexts. And so this really suggested that hyperactivation of Wnt is detrimental for enthesis regeneration. And so I just want to take a moment and summarize everything I've shown you today. I presented that the adult zebrafish is a new tendon regeneration model for the field. We've molecularly and spatially characterized different zebrafish tendon cell types during homeostasis, but as well as regeneration. We've identified conserved tendon cell populations from zebrafish, mouse to human. And also I've identified that tenocytes are a major cell source for regenerating different parts of the tendon, and that there are site-specific regenerative programs at play. In particular, that it seems like TGF-beta signaling might be more important for mid-body regeneration and Wnt for enthesial regeneration. And particularly with Wnt, it seems that the level modulation is really critical for proper enthesis regeneration, high levels being the most detrimental, and it allows us to hypothesize that potentially this is what's actually happening in a mammalian context, like an adult mouse, where potentially if non-physiologically high levels of Wnt are actually driving fibrotic outcomes. And so my current work is really focused more on trying to manipulate these pathways in mice to see if we can improve healing outcomes there. And then I'm just going to conclude on the major future direction that I'm interested in. For instance, we now have built a foundation of the adult zebrafish as a very powerful genetic comparative model that is regenerative. And so in the future, I'm really interested in merging this with non-regenerative models to perform cross-species comparative genomics and genetic studies to identify more quickly mechanisms driving fibrosis versus regeneration. And this would be paired with the generation of a new suite of genetic tools. And so with that, I would like to conclude. I really need to thank Jenna a lot for being a great mentor, especially during my postdoc. And then Hannah, Rashida, Marie, Moore, Steph, and Maddie, all people who have contributed to various aspects of this project and my funding sources. And I'm happy to take any questions.
Fantastic. Thank you very much, Stephanie. Some beautiful images there. Please do ask questions using the Q&A box. We have a question already, which is from an anonymous attendee, which is, what do you think the sources of Wnt and TGF-beta signals are?
Yeah, so that's a great question. I didn't have time to get into it. So the difficulty with deconvoluting this is that actually so I'll start with TGF-beta signaling. TGF-beta is definitely highly produced. TGF-beta 2 and 3 are highly produced within the tenocytes themselves. And they also all express TGF-beta receptors. So currently, what I'm doing and what I have done actually is I've generated genetic tools in the lab to actually manipulate these pathways in cell type specific ways. So just within the tenocytes versus the bone, for instance. So I think that those strategies are really required to deconvolute that. With respect to Wnt, actually, Wnt is not inactive in the mid-body context. I think it's just a relatively lower level of activation. And something that's interesting is what we see predominantly with the spatial data is that there might be more of a difference in terms of the activation of Wnt inhibition programs, and that we see higher levels of Wnt inhibitors like SFRPs, for instance, in the mid-body compared to the enthesis. So I think in the future, it will require finer genetic tools to deconvolute, but because there is not necessarily a very clear cell type specific expression pattern for a lot of these receptors. They're kind of just pan expression.
Okay, thank you. Rio Sasumi is asking what the key marker genes for progenitor cells for enthesis regeneration versus mid-body regeneration.
In terms of key marker genes, I would say that, yeah, there's a lot of differences in terms of the activation, like with respect to Wnt, for instance. There's some ligands and receptors that are specifically expressed only with the tendon bone attachment that you see those programs start to be reactivated again. And then like frizzled B, for instance, is one gene in particular that has known roles in directing bone regeneration and development. And so there's also differences potentially in like BMPs as well. So that's definitely still an area of active exploration. But in terms of getting at the question for whether early on versus later there are differences, I did mention that the injury responsive cell states that we have identified have actually been largely shared between both. So it seems like there's a common injury responsive program, and then later on these programs start to diverge. So I would say the earlier key marker genes are very similar and related to boosting translation, for instance, transcription, stress responses, etc.
Okay, great, thank you. A third question from John Pugh asking if you put tension on the injured tension on the injured tendon, does that affect the molecular activity in regeneration?
That's a good question. This is actually an active area of exploration in the lab. I do think it will make a huge difference. The short answer is Yes, I do think it will alter the molecular activity. There's a lot of work in the tendon field because it's a very mechanoresponsive responsive tissue that the cells do respond a lot to differences in tension and also movement, for instance. And so other people have bioengineered materials, for instance, in mammalian contexts that have increased the tension after. So making very strong adhesives, for instance, that seems to boost healing in mammalian context. So I do think that by adding tension back, it could help or some sort of cyclical tension. But that's still an active area of exploration for sure. Great area of interest. So.
Great, thank you. And I'll finish off with one question for me. So I spotted in your single cell data that you had quite a large
Sure.
immune cell contribution. And immune cells are known to play a role in sort of pro-regenerative responses in other systems. Have you been able to look at what the immune cells are doing in this
Yeah, so
during regeneration?
I haven't, but there is like another part of the lab that's looking at that. I think it's widely conserved. Something that's an interesting feature of tendons in general is that they are considered to be more immune protected. So it'd be interesting to see if there are any differences that come out, but it does seem like there is potentially a conserved role for the immune system in directing regeneration. But that's still a very active area of exploration. So, yeah.
Okay, we actually have a question from your co-speaker today, Ben, who's asking what ECM changes do you see during the regeneration? Are there changes in collagen subtypes, which has been seen in spiny mouse skin regeneration?
Yeah, there are a lot of collagens, different collagens in different parts of the tendon, actually. And so I definitely think a lot of them. Like I'm trying to think collagen 5, for instance, goes up like substantially. There's also collagen 12, which is very connective tissue like related and associated, but that also increases. There's also some specific collagens that are expressed in just different parts of the tendon that seem to change their expression potentially. So col 22, for instance, seems to kind of shift a little bit. So that'll be interesting to explore in the future because it seems like it might be more plastic. So yeah, I mean, I think that's definitely an interesting aspect. We can also talk about it more later. But we also see conserved, MMP activation to break down the matrix. So yeah, there's definitely massive changes in the ECM and collagens in particular.
Great, thank you very much.